So these were definitely not obese individuals. So, I don't know why that's all the way up there. I don't know how to fix that. Yeah. Oh well. Okay, so this is Florence. I can say her name. I have IRB approval. Just in case Mina's here. So they all completed the 10 weeks of resistance exercise training, and it was at a fairly high workload. That's going to be key here, because other resistance training studies have, or the authors have employed either lower intensities or fewer muscle groups. We did all muscle groups, 10 different exercises using major muscle groups or multi-joint movements. And it was at a moderate to high intensity, 80% of 1 repetition maximum. So, Con then maintained their normal daily activity. What did we find? Well, this is IL-1 beta production, one of the pro-inflammatory markers. Here's our HRT group before training and after training for IL-1 beta. There's our non-hormone replacement group. Here's our selective estrogen receptor placement group, and I've always wanted to follow up on that SSR. They got a big standard error there. I'm wondering what's going on. But regardless, there was no significant difference there. There was a reduction overall, and then here's our control group. So we did see a significantly reduced IL-1 beta after training compared to the control group. And this is TNF-alpha. This is, again, the really pro-inflammatory cytokine, basically the same results. Okay, so this told us that although other training studies showed no influence on inflammation, there is something going on if you use the right kind of exercise at the right intensity, whatever that is, threshold, I don't know, at least in this population. So, in general, just 10 weeks, by the way, it's a very short training period, in my opinion, reduced some of the inflammatory markers in normal weight elderly women. We'll call them normal weight. So that led us to a project here at TCU. We call it, well, we don't call it, the ladies named themselves the LOL study, the little old lady study. Except they weren't that old, they weren't that little. Anyway, I should have said that. So they were not taking any types of hormones or SERMs. Their BMIs were 30 to 40. Now we chose this overweight, or actually technically obese group, because they're at a very high risk for the metabolic syndrome and all these other things we've talked about. And they're a very understudied population, in fact, particularly in regard to resistance training. So, yeah, lots of work done in obese postmenopausal women, however, not with resistance training. So, we had an exercise group, 11 finished the study. Here's the mean age. BMI was 32.5. They did 12 weeks of resistance training this time, working, again, basically the same protocol we used in the other ladies. All major muscle groups, 10 exercises, 3 sets, 3 times a week, at a fairly moderate to high intensity there. Then we had the control group that went through some health education and social interaction to control for social interaction, because, again, we know that depression and other psychological issues can increase inflammation. So, and we also needed to control for diurnal variation, which apparently can occur as well. Not diurnal, but seasonal, because this was a 12-week training study, so it also controlled for that. And what they did, they came in twice a week for different health talks. They did some crafts. They learned how to knit. They played chess. They learned chess. Just different things to keep them active and engaged. From this one paper, we have 2 in print now, 1 in review and 1 in progress. So it provided a whole lot of data. I'm just going to share some of it with you real quickly. Okay, one of the first papers that was published reported the influence of the training on lipid changes in the blood. Okay? We observed a reduction in total cholesterol as well as your bad cholesterol, the LDL, after training in the exercise group with no change in control. You think, well, we already knew that. Well, actually, we didn't. Not all studies have shown in this population that resistance training will actually reduce lipids. We know weight loss certainly will, and consistent exercise may as well, but not resistance training. And another thing, the few studies that have been published in this population have shown conflicting results due to not correcting for changes in blood plasma levels. And this was we controlled very well. We adjusted for changes in plasma volume, and so it's made this a strong paper that would stand alone. So a paper that's in review right now shows the influence of the training on some of these inflammatory related markers. In CRP, we observed a significant decline in circulating CRP, just that which is in the blood, in the exercise group with no change in CON. We also observed a reduction in TNF-alpha circulating. That's more uncommon in general exercise literature. You can't always detect a change in this marker, but certainly we did. In the adiponectin, I don't know what happened there, this is the anti-inflammatory molecule we talked about. We did not see a change, a significant change. Maybe something was going on there, but it was not significant. So, I want to talk about this CRP. Again, it's measured in clinical medicine now. Really well-controlled aerobic training studies have failed to result in a decrease in CRP unless weight loss accompanied the exercise, either by diet or maybe through the exercise over longer terms. Again, this was a 12-week study. A colleague of mine, friend, published a study a few years ago where they combined aerobic and resistance training. They saw a decrease in CRP. Then some very well-known researchers did 2 different studies, just aerobic training, no diet. CRP did not change. So we were thinking, what is going on? It's got to be something with the resistance training. So that was one thing that led me to want to measure CRP in this study. And certainly it appears there is something about resistance training that, you know, the load, the adaptations in muscle that may be responsible for this change. Let's see. In regard to those classifications we talked about, as far as risk factor goes, based on the CRP classifications I mentioned a moment ago, the exercise group shifted from a moderate risk to a low risk, at least in terms of CRP, and that's for cardiovascular disease. So that, again, is a nice clinical finding to report to those in that area. Okay, some of the other things we measured from this study. This is kind of a busy slide, but not only did we measure some of these things just before and after training, but we did an acute exercise bout at the beginning and at the end of training so that we could look at training-induced changes to an acute bout of exercise. So let me just explain. This indicates pre-exercise, the morning of an exercise bout. This indicates post-exercise, right after the resistance training exercise bout. Then they rested quietly in the lab for 2 hours, and we got the 2-hour blood measurement, and then they came back the next morning for a 24-hour measurement. They did that both before and after training. Okay? So these first 2, the dark black and the gray, shows exercise. This would be before training, after training. Before training, after training, and so on at these different time points. Okay? This would be control before and after the intervention, before and after, before and after, before and after. What this shows is that resistance training decreased the mean, the average leptin concentration across the board in the exercise group with no change in the controls. Now, what is leptin? I just realized I didn't really mention it. Leptin is a hormone produced by adipose tissue. It signals the brain when you are full, tells you you are full. However, there's a controversy as to whether or not we should use this term. There appears to be a leptin resistance in obese individuals, kind of like insulin resistance. The fat is producing the leptin, trying to tell the brain it's full, but the brain does not respond. Okay? So it's frequently noted that leptin is elevated in obese individuals. So they're producing it. At first we wondered, why is it? You know, why are they obese? They've got plenty of leptin. Well, research has shown it appears there may be some type of leptin resistance. There's something going on. So in any case, this was a positive finding. Leptin is inflammatory as well. So we can say this also contributes to our reduced inflammation. And this is just a diurnal reduction at 2 hours. That's known. Okay, 2 more graphs. This would be equivalent to this. This is TNF-alpha. Okay. And it shows overall, rather than, actually this is the summary of it. It shows there was a main effect of training. Not between groups though. After training, TNF-alpha was higher, which is completely unexpected.
Is that bad or good? Because
Well, that's a good question. That's a good question. I'll try to explain it. This is measured not just in blood, it's not in circulation, this is in femtograms per monocyte. So what we did is cultured the blood. Not only did we measure what was going on in circulation, but we cultured some of the blood, the human blood, put into it an endotoxin, lipopolysaccharide. It stimulates the leukocytes, the white blood cells, to have an inflammatory response and to release all these cytokines. So we did this assay, and we found that after training, their blood cells responded better or more inflammatory. You don't know if that's better or not yet. And so we're like, well, what's going on? It happened in both groups. Okay? Although it looks like, if you can see the difference, that the exercise group was the group that really responded more so. It really drove that training effect. Although, this is control before and after exercise, before and after. You see it's graded there before and after and before and after. So something was happening. So we'll get to that in a second. There was also a time by group interaction. So not a training effect, but a time by group where after exercise, the exercise group had a greater TNF-alpha response than the control group. And again, on that previous study, we actually showed that during the post-exercise period, the normal weight elderly individuals had a blunted TNF-alpha response. And at that time in that manuscript, we said, hey, that's good, less inflammation. So now this is totally contradictory. These women were a little bit younger, but obese compared to our little bit older and normal weight folks. So we thought, well, it's either the slight age difference, or of course the obesity.
Melody?
Yes?
On the leptin, at that 24-hour mark, they're all back up at the same level.
Yes.
Does leptin vary?
Yes, there is a diurnal. You can see here it's kind of trending, and then by this hour it is reduced. That is a normal diurnal fluctuation.
So the exercise only had a moment or a
2 hour.
effect.
Well, this is at rest though. This is at rest, so we're not sure why these are so close. Overall, it was a main effect where leptin was lower, both before exercise, at rest after 1 week, no exercise. So, that's true. This is back up, so I can't explain. Yeah. See what I'm...
Yeah.
Okay. Good question though. Okay, so we're talking about these cultured cells and what happens when they are stimulated by the LPS came from E. coli. No, for this study it was salmonella. Salmonella makes you sick, right? Stimulates an inflammatory response. Okay. So I'm going to show you real quickly how we did this. It'll be important again. We just take the whole blood, we dilute it in culture medium, add the LPS, again that's from E. coli or salmonella, and we incubate it for 24 hours at 37 degrees C, 5% CO2, and all these squiggly lines are supposed to be cytokines. Okay, so that's our cytokine stimulation assay. We spin this down, take the supernatant, the fluid part off, and we measure the cytokine concentration. So that's how we did that assay. And oh yeah, that's our little cytokine. Now, monocytes, this is key, because we're going to get to this just shortly in an ongoing study. Monocytes are one of our major leukocytes. We have, as just a quick reminder, neutrophils, lymphocytes, monocytes, eosinophils, and basophils. So our monocytes are very responsive to LPS. So we calculate cytokine production per monocyte in the blood because they're a big LPS responder. So This is interesting. There are different levels of maturity of our monocytes, though. You think of monocyte as a monocyte? No, you have classical